Your teacher Irfan Majeed but from Nahama. Today I would like to discuss some basic functions of the nephron, as I have already uploaded one video about the structure of the nephron. So here we go. As you can see on the screen, nephron uses some basic functions for the formation of the urine, which are filtration, reabsorption, secretion, and excretion. Now, I have seen most of the biology teachers in the YouTube, they just explain filtration and reabsorption and job done. This is how urine is formed. Well, not quite right. See, we can understand here in a better way. We will look how the blood is filtered at the level of the renal corpuscle level, and then we will understand how the urine moves forward. So here we can see on the screen the blood enters the kidney via the renal artery. This is a large artery and the branch of aorta. After entering into the kidney, it gets divided into many small divisions until it reaches the smallest one that enters the nephron. This is the afferent vessel that brings the blood into the glomerulus, and this is the blood vessel that takes the blood out of the glomerulus, that's known as efferent vessel. In between these afferent and efferent vessels, there is a jumbled mass of capillaries known as glomerulus. And that we have already discussed earlier. It has derived from a word glomer, which means a ball of thread. Glomerulus is a tuft of blood vessels that is located inside the Bowman's capsule. It greatly increases the surface area for filtration that comes in contact with the Bowman's capsule. This is where the first mechanism of the filtration takes place, filtration of the blood. We can understand the filtration when we come to know about the glomerular filtration barrier. So we need to understand it. If you take this part of the renal corpuscle and look at its microscopic view, it would look something like this. So this is the blood vessel wall of the blood vessel. Then this is the basement membrane, and here are the cells called the podocytes. And there is a gap between the podocytes. These are the special types of the cells. That gap is called as the filtration slits. Together, these three layers form the glomerular filtration barrier that acts like a filter paper. So we have blood coming in this blood vessel that gets filtered through this glomerular filtration barrier. And then the glomerular blood pressure, that is actually the driving force for the filtration here. And the filtrate moves into the Bowman's space, and only about 1/5 of the plasma is filtered here, and rest is passed on. The things that do not get filtered, they are red blood cells and some larger molecules like larger proteins. They are not filtered here. Also, one important thing you should note here, that is the difference in the diameter of the afferent and efferent blood vessel. Afferent has larger diameter and efferent blood vessel has smaller diameter. So that's why the pressure is created inside the glomerulus, which provides a driving force which makes this filtration possible. The kidneys can adjust the rate of glomerular filtration, so because of some hormones also, and because of the diameters can be adjusted, these two diameters. Now we will look where this filtrate moves. The plasma that gets filtered into the Bowman's space, that is known as the glomerular filtrate. Now next it moves into the renal tubule. It moves forward in PCT and then into the loop of Henle and then into the DCT. So it has some essential products also in it, in the filtrate, nephric filtrate, like NaCl, glucose and all, which we do not lose. Approximately there are 2/3 of the filtrate salts in it, and glucose is also there, which we don't want to lose because glucose is a hard-earned stuff that we have got from our food We also don't want to lose sodium chloride and amino acids also. So it is achieved by a method of reabsorption. So here the whole length of the renal tubule is surrounded by blood vessels, which are known as peritubular capillaries, where whatever gets reabsorbed here, that is passed back into the blood circulation, it is known as reabsorption, but not absorption, because the same nutrients have been absorbed into the blood once, in our gut, in our intestines once. So here we have got these renal tubules have protein channels. Through these they get reabsorbed by the blood. Now about 70% of the sodium chloride gets reabsorbed in the PCT, proximal convoluted tubule, and 70% of the water, more than 95% of the glucose and 95% of amino acids get reabsorbed in the proximal convoluted tubule. Now the next nephric filtrate passes into the loop of Henle. Loop of Henle has some role in the reabsorption of the important molecules like sodium, calcium, potassium, magnesium. As you can see, 25%, calcium 25%, potassium 20%, and magnesium also 60%. Loop of Henle has a major role in the concentration of urine, in making the urine concentrate. But it is taught at higher levels also. But I will try to simplify it here as much as possible. So please pay close attention. This is the section of kidney, and you can see how the structure of kidney is divided into cortex and medulla. The inner medulla of kidney is a very important region, as it contains higher concentration of salts as compared to the cortex of kidney. This higher concentration of salts is made possible by the loop of Henle, which you can see here. You can see how this loop of Henle is present inside the medulla of the kidney, and rest of nephron is present in the cortex. The loop of Henle has descending limb and ascending limb. If you can look at the ascending limb of loop of Henle, it contains special ion channels through which the sodium and chlorine, they come out, and it makes this region more hypertonic. The ascending limb, however, does not allow water to move out, as it has no channels called aquaporins for the water to move out. But these types of the channels allow the water to move out in the descending limb. They are actually present in the descending limb, these aquaporins. That makes it freely permeable to the water. Now this is all salty region here, and the water will move out in the descending limb in the renal medulla, and then eventually it gets reabsorbed into the blood vessels. This is the reason why loop of Henle is so long, so that that water will get time to secrete out, and that is why it dips pretty down into the salty portion, into the medulla. So this is the major mechanism of gaining back of the lot of water that gets filtered up in the renal corpuscle. After the loop of Henle, the water passes into the distal convoluted tubule. It is this part of the renal tubule where we have the reabsorption of ions like calcium, sodium, and it just reabsorbs more things which we don't want to lose at the first place. So here now you can see in distal convoluted tubule, another mechanism of the urine formation takes place, which is called the secretion. Secretion is the opposite of reabsorption. In secretion, the waste ions and hydrogen ions, they pass from capillaries into the renal tubule. So this is the final step in the formation of the urine, during which walls of the tubule, walls of the blood capillaries actively remove the harmful products, there are, you can see here in the diagram, and these once they move into the renal tubule, they can then go towards the collecting tubules, like hydrogen ions, potassium ions, ammonium ions. They get out of the blood vessels into the tubule here. This is the secretion. Some secretion also takes place in PCT region, where the substances like urea, uric acid, creatine, ammonium ions, and some other products also, they get secreted from the blood into the PCT. Now, then at the end here, we have our filtrate which is ready, that has been processed. A lot of water has been taken out. It is very much concentrated. We have reabsorbed lots of salts, electrolytes, all the glucose, amino acids back into the blood. Now this is the urine, which is now present in the collecting duct. There are so many collecting ducts which join the same duct then, like you can see here by arrows. Now collecting duct is very important from two aspects. First, it dips back into the medulla, and then second, because anti-diuretic hormone that acts on the collecting ducts, and it decides the concentration of the urine. It is a whole different topic, it's also called as the vasopressin, secreted by the posterior lobe of pituitary gland. Now this urine is drained into the renal pelvis, and renal pelvis is in turn connected with the ureters, and then ureters are connected with the urinary bladder, and urinary bladder with urethra, and like this the urine is excreted out. Thanks for watching me.